CMI travels all over the world and does these messages, these awesome teachings, and all they ask for is a love offering. So we're going to be taking a couple of those this morning. I'd also like to announce our co-sponsor is Noah Webster College. I don't believe John is here at this point. He wanted to say a couple of words, but Noah Webster College is a local source that teaches creation science classes. If you're interested, you can find them online. John and Terry, are you guys back there? Also, a reminder, these little yellow slips up front. These are just a little sign up for a drawing we're going to have. We have some CMI materials that we're going to be giving away at our next meeting. We didn't want to compete with their sales here tonight, but we did purchase some of their materials, and what's left, we'll probably be purchasing some of that. We'll have it available in our lending library as well as on our tables at our regular meeting. So if the ushers could come forward, please. We'll be doing this drawing some point after lunch today. Father, we just pray that you would bless this offering, Lord, that you would bless CMI for their amazing ministry. Father, as they travel around the world and teach the truth of your creation, pray that you bless this offering in Yeshua's name. Amen.
Ooh.
Jonathan.
Sure, thank you. Go ahead.
One more thing. Right after the offering, if we could pass the white clipboards around, these are sign-up sheets for CMI.
These ones, yes. Okay, shall I start then? Yeah, thanks. The point about the white sheets is to get people connected with a ministry so we know what articles are coming up in the future. It tells you some of the classic articles from our archives, and it also tells you when a CMI speaker is in your area, because I think we find a lot of people come to as visitors to a church because they've read about us in the newsletter. And just to remind us why we do what we do is that there is a big Christian youth dropout rate. Now, to work out why this should be, we have a very short video called Fallout, and the disc is actually on the table there. And we interviewed various students about whether they actually went to church and they still continue in church. It's interesting that there are a number of those who were brought up in the church but are no longer attending. And they were asked, were you ever taught about creation in the church? No. So they had no answers to what was actually being taught in the schools and universities. But there were other people who, when we asked them, they continue in church, and they all said that they were taught about creation and how to defend the faith. It really makes a big difference what we believe, what we've been taught. Because we are taught in Romans, the invisible things of Him from the creation of the world are clearly seen, being understood by the things that are made, even the eternal power and Godhead, so they are without excuse. So that means Darwin and Dawkins have no excuse for their unbelief, because the creation is so plain. But when we go back to what the definition of evolution really is, we see that origin of life is part of it. I know the evolutionists, a lot of them don't want to even admit they have a problem, so they pretend that origin of life is not part of evolution. It's abiogenesis, that's their favorite term nowadays. But according to the general theory of evolution, that's the theory that all living forms in the world have arisen from a single source which came from an inorganic form. So it's the non-living chemicals to first life is part of evolution. It's often called chemical evolution. That's the common term for this theory of life from non-living chemicals. So what I'm talking about definitely is part of the materialistic worldview. They have to believe, if they don't want to believe in a creator, they have to believe that somehow non-living chemicals formed into a living cell. Now, I'm talking about design of things, so let's look at what design is about. You see, we make things ourselves. Okay, how do you know an aeroplane was designed? If you didn't see it being manufactured, how would you know it's been designed? Well, I'll say one thing you might see is loads of correct components. But you realize an aeroplane is made up 100% of non-flying parts. So how am I going to get home to Atlanta? Well, the answer is they're organized correctly. So not only do we have correct components, but we also have correct organization. And organization requires an organizer. See, this is not difficult science. This is observational science, that wherever you have organization, there's an organizer. So what's the alternative here? See, evolution basically says if you have matter plus time plus energy, you get organization. So I'm illustrating this with an experiment here. A tornado going through a junkyard, tornado being energy, junkyard being matter, and as much time as you want, and you get an aeroplane from that. So how realistic is this? Not really, okay, I imagine. But what if I told you it's the other way around? It's a tornado hitting the aeroplane and turning it into a junkyard. Is that better? But do you know why it's better? There's a very important principle here, is there are many more ways of being a junkyard than being an aeroplane. That's all it is. So when you add the energy and the time, you go to the more probable state. Just different varieties of junkyard. No matter how long you leave it for, time and energy don't help you. It just goes to different varieties of junkyard. Other than that, its principle is very wide-ranging. So tell me, do you have to tell your children to mess up their rooms? Again, the reason is there are many more ways of being messy than being tidy. So if you want a tidy room, you can't let the kids just use their energy. They have to plan to put things together in a tidy way. Now, of course, the Richard Dawkins of the world will say, I've overlooked something. I've forgotten about natural selection. Well, actually, no, I haven't. Because here is what natural selection actually is about. Here's Theodosius Dobzhansky, was one of the leading evolutionists of the 20th century, and he said natural selection is differential reproduction. To have natural selection, you have to have self-reproduction or self-replication, and at least two distinct self-replicating units or entities. A pre-biological natural selection is a contradiction in terms. You see, natural selection says survival of the fittest and death of the unfittest, but it really is this thing is fitter than this thing, so this thing leaves more offspring. So to have natural selection, you must have reproduction. Therefore, natural selection can't explain the origin of first self-reproducing things. All you have are matter and time and chance. Natural selection can only start once we have something that can reproduce. And it's interesting that what I'm going to talk about was very important for the philosopher Antony Flew, who was for decades the leading atheist philosopher. Now, we've got a book, Christianity for Skeptics. Now, an earlier edition of that, some years ago, quoted Antony Flew as a representative atheistic philosopher. But in the new edition, we had to update that because he became a theist, a believer in a God. Unfortunately, he doesn't seem to have become a Christian, sad to say. But one reason that convinced him is what he's saying here, that Richard Dawkins overlooks the fact that Darwin pointed out that his whole argument began with a being which already possess reproductive powers. And this is the creature, the evolution of which a truly comprehensive theory must give some account. Darwin was well aware he had not produced such an account. It seems to me that the findings of more than 50 years of DNA research had provided materials for a new and enormously powerful argument to design. So just considering the complexity of even the simplest living thing it ruined his atheistic faith, totally destroyed his atheistic religion, thanks to understanding the complexity of life. Now let's go back to that aeroplane, and we see again, aeroplanes are not living things. Living things are much more complicated than man-made things because they can make copies of themselves. See, a Boeing jet doesn't have baby jets. But living things make copies of themselves. Your children are sort of copies of you, okay? And now your body's made up of about 30 trillion cells, and all your cells are making copies of yourself, of themselves. That's how your body grows and repairs itself. Now it's interesting, the 30 trillion human cells is based on a 70 kilogram man, based 150 pound person, okay? It's interesting, there are probably about 40 trillion bacterial cells in your body. So you have more bacterial cells than you have human cells. And they're good for you, okay? Hence the term probiotics, okay? Those are good bacteria. And this also leads to what the appendix is for. It is not a useless organ, a vestige of evolution. What the appendix does is a storehouse for the good bacteria. So if you have an intestinal infection and then it's flushed out, the bacteria go. But what happens, the appendix stores the good ones so they can repopulate the large intestine. So the appendix is a very important function, and people who lose it, they lose the safe house for their good bacteria. And now they're talking about, because it's a useful organ, to try to treat appendicitis by injecting antibiotics instead of chopping it out, because they're realizing how good it really is. Unfortunately, here's a case where evolution has harmed medicine. By dismissing the appendix as a vestige of evolution, people have been harmed by that belief. When surgeons would just go in, oh, they've got something there, while we're at it, let's chop the appendix out, it's useless. No, sorry, it was not useless. It was a medical malpractice, thanks to evolution, that was. So again, but living things are enormously more complicated than any man-made machine. So it can copy itself. See, a Boeing factory can make jet planes, but it can't make you a new Boeing factory. A photocopier copies documents, but doesn't make you a new photocopier. But living things copy themselves. So how did they arise? Well, again, the evolution idea is matter plus time plus energies, which I will test with a frog in a blender. And I turn the switch on, I'm adding energy to the system, right? So and the result is a frog smoothie, okay? Now, you'd think if I left this going for millions of years, a frog will hop out of here again. See, matter and time and energy is not enough, is it? And by the way, the reason, again, there are many more ways of being a smoothie than being a frog. See, it's a useful principle, that. And by the way, no real frog was harmed in the making of my talk. Now, another thing you can think about this, if you canned this and sterilized and sealed this, this would be safe to eat for years. You would not get food poisoning, because food poisoning comes from bacteria, which are microscopic organisms. They give you food poisoning, but they're living creatures. But if you can and sterilize it, even though the canned frog smoothie or canned Georgia peaches or sardines or whatever you like have all the components of living things there, but no life will ever form in there. Unless the seal got broken and the germs came in from outside and contaminated it, but it's not going to come from within. And this is a very life-saving law discovered. It's called the law of biogenesis. Louis Pasteur discovered, and then Joseph Lister based antiseptic surgery on Pasteur discovery. What Lister realized is that before him, surgery was as dangerous as the condition it was treating because of infection rates. But what Pasteur and Lister realized is that the infection did not come from within, it came from outside. It could not come from within. So what you do is you have an antiseptic spray and you stop the germs coming in from outside and the wounds don't get infected. So that's saved millions of lives. So this is a law of real science. The law of biogenesis says life comes only from life. Evolution says life comes from non-living chemicals. So evolution is anti-science. So let's go into what do we need for living things, even simple ones. And I'll go through these one by one. Well, first I've actually explained you need reproduction. Now, reproduction is transmitting the information that makes you who you are. That's an important thing. Now, the information in living things is in this famous DNA molecule, or National Dyslexia Association. Actually, it stands for deoxyribonucleic acid. This is actually a chemical message molecule. It has the instructions to make you who you are. And the instructions are in a form that it can be copied to the next generation. So you didn't really get your mother's eyes or your father's ears. The instructions were copied to you. When the fertilization happened, that's when you got half the DNA from your mother, half from your father, and became a new living being. And it's a scientific fact that life begins at conception. This is not a religious viewpoint, it's a scientific viewpoint. So Planned Parenthood is anti-science by pretending that the babies aren't human. Well, actually, they don't pretend they're not human because they want to sell their parts, so they have to say they are human after all, after we've chopped them up. Okay, but yeah, real science is life begins at conception. Now, DNA is a very interesting molecule, deoxyribonucleic acid. Now, it's a little bit of basic chemistry here. An acid is defined as something that gives off a trihydrogen ion or a proton. Now, the proton is positively charged, so when the acid gives the proton to something, what's left behind is called the conjugate base. And because it's given off positive charges, what's left behind is negatively charged. In fact, DNA is a stronger acid than vinegar or lemon juice. And that's actually a design feature because the negative charges actually repel certain chemicals that would try to destroy, would react with DNA. But the negative charge repelled a lot of those. So it actually makes it more stable than it would otherwise be. And also we have these bases here. These are called bases. These are flat molecules. And in the DNA helix, they actually pack upon each other. So there's actually a chemical thing called pi stacking. And it's not pi as you eat, it's the Greek letter pi. It's a type of chemical bond, okay? And the pi stacking helps to stabilize the double helix. They're flat molecules. And also they bond to each other. And the bonding is unique. So the adenine bonds only to the thymine and guanine to the cytosine. And this is how reproduction occurs, because when DNA makes a copy of its message, it makes a sort of photographic negative. So wherever it has an A, the daughter strand must have the T. So you've got exact digital reproduction of a DNA message. And here's a video from our DVD called Evolution's Achilles Heels, which we produced ourselves, about what's actually. This is a DNA double helix, and we're going to try to magnify it and analyze. You see how the helix is coiled upon coil, so you've got super coiling, you've got extra coiling upon coiling, and then it's arranged in the cell in a certain part. And it's also, the DNA actually has instructions for how it is arranged in the cell. Because, see, every one of your cells has all the information to make you who you are, but most of it is switched off. See, your eye cells have information for your thigh bone, but you don't want your thigh coming out of your eye socket. So the thigh information is switched off. in your eyeball, okay? So only the eye information is read. And the cell makes sure the eye information is accessible for the reading machines. And there's no point, if the bone information is switched off, let's put it deep where we don't need it anyway. So the DNA actually encodes for its own arrangement in space and time. Really quite amazing what it does. And as I say, DNA has the information that can copy itself. But DNA doesn't really copy itself. It requires a lot of machines for DNA to copy itself.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
So you see, there's enormously complicated machinery required for DNA to copy itself. But here's a sort of chicken and egg type problem, that DNA codes the instructions for its own copying machines. But unless you already had copying machines, the instructions couldn't be passed on. So you have to have both the DNA instructions and the copying machines, otherwise life could not even get started. There's an enormously complicated copying machinery required for even the simplest living things. Now, it's interesting that DNA is the molecule of life, because I've explained how there are certain features that confer some stability, but even so, DNA is actually very unstable, despite all that. There's a general belief that DNA is rock solid, but actually DNA is highly reactive. On a good day, about 1 million bases in the DNA in a human cell are damaged. Now, you've got about 3 billion bases or letters, DNA letters, and every day you have 1 million damage in each of your cells. So you'd be a mutated mess really quickly. And this actually was important for awarding the Nobel Prize for Chemistry 2 years ago. These 3 men were awarded that prize, and here's what the Nobel Prize website said. In the early 70s, scientists believed that DNA was an extremely stable molecule, but Thomas Lindahl demonstrated that DNA decays at a rate that ought to have made the development of life impossible. See, DNA is so unstable, so how can we cope with it? He was motivated to study, to find repair machines. He realized that cells had to have repair machines to undo the damage that happens to DNA. Otherwise, life couldn't exist. And so he was motivated to try to discover some of these repair machines. And the other two scientists discovered different varieties of the repair machine. We've got loads and loads of repair machines, and everything from bacteria to humans have a lot of different repair machines. Otherwise, again, we'd have a problem. But the thing is, there were no repair machines in the primordial soup, if there ever was one. In fact, there's no evidence. There's not the slightest evidence that the primordial soup ever existed. Even though it's an article of faith for evolutionists that such a thing happened, there's no evidence that it existed. And certainly, you would not have repair machines in a primordial soup. So DNA would break down quicker than it was formed. So a huge problem. And the other thing is that DNA codes for the instructions for its own repair machines. That's an amazing problem because, again, if you didn't have repair machines working almost perfectly, the DNA instructions would break down, which means the next generation would have even worse repair machines, which would do a worse job of repairing the errors, which would make it even worse set for the next generation of repair machines and so on. So it's what's called an error catastrophe. And you can mathematically work out that if you have something with n letters, you're only allowed to have one in n mistakes. Otherwise, the thing breaks down. And this is actually part of what's called the eigen paradox. You see, we can measure the rate of replication by itself without any repair machines. And in RNA and DNA, at best, it'll be 1 in 100, which would mean you can't have more than 100 before the thing falls into error catastrophe. But 100 letters is not enough to code for repair machines. And there have been various attempts to solve the paradox, but none of them are very satisfactory. Because if you have something large enough to code for repair machines, it's going to be more prone to mistakes. So if it was 1,000 things long, you'd only allowed 1 in 1,000 errors before collapse. In fact, some antiviral drugs, what they do is try to increase the error rate so that the viruses basically mutate themselves to death. So you need a very high fidelity copying right from the start. Otherwise, life couldn't get started and natural selection couldn't get started. Now, this is actually an interesting thing. The instability of DNA has implications because it has been found in dinosaur bones, which is an incredible surprise. Because the dinosaur bones were alleged to be 68 million years old, and there's a certain test they use. There's several tests they use for DNA, and it's a fluorescent thing that actually lodges it in the double helix groove. So it means to get the positive signal, it had to have a double helix, which means you had to reasonably intact DNA for that probe to give a signal. However, measurements of DNA instability show this could not happen. See, the real science, discovery of DNA, there's also real science about how long the stuff could last. Now, even if you froze it to -5 proper degrees, or 23 of your degrees, it's below freezing, right? And the maximum survival time is under 7 million years, and that's time to complete breakup into single letters. And I pointed out here, for this probe to work, there had to be intact DNA joined together into the helix. And yet, in a tenth of the supposed dinosaur age, DNA would have completely broken down. And this is if it's freezing. Now, dinosaurs were meant to have lived in tropical climates, which means the rate of breakdown is much faster. So if you go up to, say, a mild, warm day of, say, 77 Fahrenheit, the DNA would last just over 20,000 years before it's completely broken apart. And also in the real world, places like Montana, which have those dinosaur bones, you have thaw and freeze cycles, which would actually break up DNA pretty fast as well. So everything about it is against the millions of years. And again, this is real science of how fast DNA breaks down. And these actually might be a bit generous to the evolutionists, these time scales here. In reality, they probably are even less stable than these numbers imply. So finding DNA is a good evidence against the millions of years for dinosaurs. But also is an intractable problem for trying to explain origin of life. The main chemicals of life are very unstable and just would not last millions of years. Now, another thing with DNA, it's a very long and skinny molecule. I mean, it's only a few nanometers thick, the helix. But in fact, the biggest chromosome is about this long or so. It's only a few nanometers thick. You can't see it without an electron microscope, but it's this long. And in fact, if the DNA in your cells were put end to end, it would be about 2 meters long. So you have a very, very long, skinny, and sticky molecule, and that provides some problems. I mean, some of you guys might be old enough to remember the telephones with the cords, the coils, and how they got tangled up easily. Okay, that's why people go to the mobile phones to avoid the cords tangling up. And that's a problem for DNA. So how do living things solve that problem? I'll show you what happens.
Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase 1 enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase 2 enzymes do the same thing but with both strands of the double helix. Topoisomerase 2 cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase 2 can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
So whenever the DNA is separated, it introduces one twist, and the topoisomerase has to run behind that helicase to untangle the mess that's produced by the tangling. And without these enzymes, life cannot exist. Bacteria have these enzymes. And in fact, some antibiotics, the fluoroquinolone type of antibiotic like Cipro or Levaquin, what they do is work on the final step, which is putting them back together again. So what happens, the topoisomerases work, they chop the ends of the DNA, but don't put it back together again. So the DNA is torn to shreds, and the bacteria die. Because this shows how topoisomerase must have arisen in a complete form. There's no point a half evolved or a third evolved topoisomerase, because you've got 3 things it has to do. It has to chop, it has to rearrange, and it has to put back together. There's no point having an enzyme that just chops and doesn't put it back together again. That's what happens with the antibiotics. It stops the putting back together again. It's not enough. It just does more harm than good. So you have to have everything working together. Otherwise, this is a totally useless thing. So natural selection can't produce this gradually, because unless it's all working together, it doesn't work at all. And furthermore, natural selection can't even be used here because it's required for first life. So you can't use natural selection to explain the origin of topoisomerases, because you wouldn't have it unless you already had these enzymes untangling the DNA. And the usual chicken and egg problem works here, that DNA encodes the instructions for its own topoisomerases. But you could not read those instructions unless you had these enzymes already there. So you need both the instructions and the machines. Otherwise, life could not even get started. And now I should talk about what the evolutionists, most of the people working on the origin of life, they try to think that there may be some molecule that can do both things. It can hold the information, but also do some of the work that proteins do nowadays. The enzymes that I've shown you, they're made of proteins. But maybe they think there's something that can do both the protein work and the reproduction work, and their candidate is a molecule rather like DNA. It's called RNA. But one obvious problem is that the RNA molecule has this very important difference. The DNA deoxyribo, here's ribo, so remove an oxygen is deoxyribo. Now the point is, these groups here are reactive sites. This is actually a stabilizing part here. This is reactive. So that means that RNA is far more prone to reactions. It even reacts with itself to link the different parts. So it's a very much more unstable molecule in DNA. And yet I've shown you DNA is incredibly unstable. RNA far more unstable. So the idea of RNA appearing in a primordial soup is chemically preposterous. It is just too unstable. And even the building blocks of RNA, like ribose, quickly disintegrate. And it's worse in high temperature. Like some people want to have life beginning at these hydrothermal vents, but that would be the worst place to get these molecules because they break up more quickly. Now, there are living things that live there, but they have special repair machines to cope with that damage. But you can't have those repair machines unless you already have living things. So you can't use that to explain the origin of first life. It's a very unstable place for chemicals to try to accumulate. They break down. And here's an interesting admission. This is a paper written by a German biochemist who's working in New Zealand. And he gives you these problems with RNA. One is that RNA is a very complicated molecule. It's not a simple molecule, and it's unstable, and it doesn't do a very good job of the things that proteins can do. Proteins are far more versatile. They do far more things. RNA is not very good. And you notice the title of his paper here, The RNA World Hypothesis, the worst theory of the early evolution of life, except for all the others. See, the thing is that's what they want to believe in materialism, a materialistic origin of life. This is the best they got, but it's got problems. The only advantage it has is everything else is even worse. Okay, now let's look at another thing that living things require, and that is the information, the minimal amount of information that's needed. The simplest living thing that reproduces itself is called mycoplasma. And see, this is a problem, though, because this is a parasite. This can't really get all the things it needs on its own, so it has to be a parasite. It's too simple to survive on its own. See, viruses I don't count as living things because viruses have to hijack reproductive machinery from other cells. They can't reproduce by themselves. So this is the simplest, genuinely living thing. And it has over 480 genes, or you might say it has 600 kilobytes of information. That's the simplest living thing. But the information must be incredibly compressed. Because we're trying to duplicate on a computer simulations of what happens when mycoplasma divides in two, it reproduces. It took over 100 computers running 9 to 10 hours to duplicate it because they had to produce about 0.5 gigabyte of information during that reproductive step. So the information here must be incredibly compressed, very incredible data compression software here. And also, DNA has the highest information density of anything that we know. You see, DNA Valley would leave Silicon Valley in the dust. If you want some figures, the information density in your cell is about 1,000 terabytes per cubic millimeter. See, what size is your hard drive or your solid state drive? Okay, it might be a couple of terabytes. And it's a very big thing, but a cell is 1,000 terabytes in a cubic millimeter. There's nothing that we have that's even remotely like God's information technology. Now, of course, humans have far more information. We have 5,000 times as much information as the mycoplasma. We have about 3 gigabytes, again, highly compressed information. If the information was written in ink molecules on paper, it would take 1,000 Bible-sized books to write it out. Mind you, only 200 times the size of the IRS tax code. The point is that the information in the books is written with ink molecules on paper, but the information did not come from the ink molecules. There's nothing in the chemistry of the ink to make it write the messages of a book. That required an author to arrange the ink molecules into letters and sentences, words and sentences and paragraphs. But the information did not come from the ink. And exactly the same way, there's nothing in the chemistry of a DNA to make it write out the messages of life. So just as the book's information doesn't come from the ink chemistry, neither does the information of life come from a DNA chemistry. In both cases, the information comes from outside the chemistry. It doesn't come from within the chemistry. Now, another thing, for a book, you have to understand the language. See, my books aren't much use to you unless you understand English. For instance, if I gave you a gift, you'd be happy, right? Because it means something you get for free. Now, if you were Germans, would you be happy if I gave you a gift? Why not? It means poison in Germany. I'll prove it. Here is what they used in the Holocaust to exterminate the Jews, at least in Auschwitz and I think a couple of the camps. Other camps use different things. I'll talk about that after lunch. But this is called Giftgas. It's the hydrogen cyanide used for exterminating Jews in Auschwitz. Okay, so gift means poison, which means you have to have the right language. Otherwise, you get the wrong messages, which means you have to have decoding machines. I'll show you what actually happens when the DNA message is decoded to form proteins. But as I said before, there are more than one code. There's also code about how the DNA packs into a cell in space and time. So DNA in your cell is more like a book you can read in English and in German and backwards. You have not just one code, but multiple overlapping codes. And here's what happens when the codes get read into one of the codes into proteins.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a two-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperonin. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperonin achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
So again, we have incredible machines required to decode the DNA into the proteins that it's coding for. So without these machines, life couldn't even exist. It couldn't even get started. Now, it's interesting, again, we can look at what antibiotics do, and some of them destroy steps in this process. Like the streptomycin antibiotic, it binds the ribosomes, so the bacteria can no longer make proteins. The tetracycline antibiotics, they do something a bit different. They stop the connector molecules, the tRNA, the transfer RNA molecules from binding properly to the ribosome. And yet another type of antibiotic called the macrolides, they stop the protein chain growing. So any one of these steps is ruined, the whole thing collapses, which shows that everything must have been coordinated for even the simplest living thing to arise. And again, we have the chicken and egg type problem here, that the DNA information is useless unless it's decoded, but the instructions to build the decoding machines are on the DNA. So which came first, the decoding machines or the instructions to build them? And the decoding machines must, again, be almost perfect in their decoding. Otherwise, the next generation of decoding machines is not made properly, which does a worse job of decoding, which does an even worse job of making the next decoding machine, and so on, until we get the error catastrophe again. So lots of different things are needed for living things. Now, I want to go through yet another thing, what is needed for living things. Because what they try to do is work out how simple could life be. So let's take the simplest thing we have and see if we can disable some of the genes and cripple it to see what it can do without and what it can't do without. And it seems that the minimum genome has to be about 387 protein coding genes are required for even a simplest living thing, and they're doubtful whether this could even survive. It seems to be so simple and degenerated that it couldn't survive. And it's interesting that people analyzing the simplest forms of life think they have degenerated from more complex forms of life. That's part of the fall, the deterioration after the fall. So the first life had to have been more complicated than this one. So let's do a probability calculation. I'll go through some assumptions here. First of all, 10 to the power of 80 atoms in the observable universe. So 1 followed by 80 noughts, or zeros as you call them here, right? That's what 10 to the power of 80 means. So it's a big number. And supposing we've got very fast chemical reactions, so we have 1 trillion reactions per second. And then if we allow the Big Bang, which I don't agree with, but let's assume it for the sake of argument, there are 10 to the power of 18 seconds in the Big Bang. Now, let's multiply all these together. And then when you multiply such numbers, you add the exponent, add the index on it, okay? And so therefore, the product of these is 10 to the power of 110. Now let's look at what we have for living things. There are 20 types of amino acids used in most living things. Those are the letters of proteins, are the amino acids. And we have 387 enzymes for even the simplest possible life. Now, enzymes have to be incredibly very specific, because they're supposed to do one particular thing, like untangle the DNA and put it back together again. So they're very specific things. So there must be some constraint on what could actually produce an enzyme. So how many amino acids do you have to have for the enzyme to work? I'm going to be very generous to the evolution and say that we only need to get 10 of these right per enzyme. In reality, it's far more than that. Enzymes are far too specific. You need a lot of things to go right and not much that can go wrong. But even if you assume these numbers here, you work out the probabilities here, And it turns out to be 10 to the power of minus 5,000. Now, these numbers are just too big to imagine. So the easiest way I've found to sort of work out how enormous these numbers are, think of your bank card. Your credit card, it has a PIN. People really shouldn't say PIN number because PIN means personal identification number. So PIN number is personal identification number. Or HIV virus, ATM machine. And with a term for this, it's called RAS syndrome, or redundant acronym syndrome. Okay. Now, okay, so you've got your credit card. You've got a 4-digit PIN, okay? That means every digit can be 10 digits, from 0 to 9, okay? So every digit is a power of 10. So when you have 4 digits, you've got a 10 to the power of 4, or 10,000. So you've got 1 in 10,000 chance of getting that right. And the bank will tell you, if someone got it right, they will say that either you left it lying around somewhere, or else someone stole it. They phished it. They used some sort of scanner, and that's how they got your PIN. See, without that, they know it couldn't be cracked by chance. So every number you add to a PIN, you see, a 5-digit PIN will be 10 times harder than a 4-digit PIN. And a 6-digit PIN, 100 times harder. But what we're talking about here is like trying to guess a 5,000-digit PIN first go. That's the sort of chance we're talking about to have life from time and chance. And that's allowing for the whole universe to be full of chemical soup. And allowing the billions of years that they believe the Big Bang allows you. But in fact, just to show you how generous I've been, here's a typical enzyme called calmodulin. It's used for calcium modulation, okay? And it seems that 142 enzyme of the letters have to be exactly right. Everywhere we find calmodulin, 142 letters are right. So it just shows you how getting this by chance is not an option, but getting it by natural selection is not an option because you haven't got reproduction yet. So all you have is chance. Now let's look at yet another thing we have here, that life uses energy. See, you eat your food for the energy. But in the cell, the energy is this stuff here called ATP, or adenosine triphosphate. And all living things need this for energy. In fact, your body makes its own weight in this stuff every day and consumes it. Cyanide kills you. The gift gas I showed you, that works by stopping ATP production. And it's deadly. But now we know it's made by the tiniest motor in the universe.
This animated sequence shows the ATP synthase enzyme in operation. The animation is based on an incredible series of scientific discoveries. Only the colors show artistic license. ATP, or adenosine triphosphate, is the energy currency of the cell. ATP is produced by a tiny molecular rotary motor, rotating at up to 7,000 RPM. These are so small that 100,000 would fit side by side in a millimeter. A current of protons drives the motor, unlike man-made electric motors which use electrons. This portion of the enzyme is where adenosine diphosphate is combined with a phosphate ion in the presence of a catalyst to produce ATP, which is then released, making way for the next cycle. A top view of the enzyme shows the sequential operation. Almost every biochemical process in your body requires ATP. Such a nanomachine exhibits all the characteristics of super-intelligent design. ATP is vital for life, and many of these motors were needed before the first living cell could exist. An evolutionary impossibility.
Actually, when I was in California a couple of weeks ago, I gave a talk about this, and it turned out that one man in the audience who's a creationist has actually published papers on this motor very recently. And he told me you can disable one of the many protein components of the machine, and it totally disables the motor. which shows that everything has to be very well organized. No part can be missing, otherwise the motor won't work, and therefore energy couldn't be produced. So incredibly important stuff, this. And also there's an antibiotic called oligomycin that actually blocks the bottom motor from spinning. So we can actually know what happens when these things don't work, the living thing dies. And going back to what we saw here, that we have the DNA requires decoding machines, which are themselves made on DNA, but the decoding machines use energy, which means most of them use ATP, but to make ATP, you need the instructions on the DNA read by decoding machines using ATP made by the motors. So it's what do you call a three-way chicken and egg problem. That's what we have here. So what can I say the next time thing about this is even things like enzymes are incredibly complicated things. They speed up reactions. In fact, one of the enzymes required for DNA decoding into proteins acts as a double sieve. It's a very intricate thing because what it does, it sieves out the things which are too large. But what about the things that are too small? It had an other sieve to get rid of the things that are too small. So you need this enzyme so you can link the right three-letter code on RNA to the right letter of the protein. So you're having these incredibly complicated enzymes required for DNA reproduction. And some of them speed up reactions by an enormous amount. Like one reaction that's vital for life would take 78 million years without this enzyme, but it's sped up by 10 to the power 18 times. That's incredible. You see, the thing is, we haven't got 78 million years in a primordial soup because the stuff is so unstable. So we've got to do it quickly, otherwise the thing breaks down. And another one speeds up by 10 to the power of 21 times. So without this reaction, without the enzyme, the reaction would take many times the Big Bang age of the universe. But the enzyme enables it to happen in a fraction of a second. And here is one of the experts on this. He said, without catalysts, there'd be no life at all, from microbes to humans. And here he makes a really incredible blunder, which I hope you can now work out what the blunder is. It makes you wonder how natural selection operated in such a way as to produce a protein that got off the ground as a primitive catalyst for such an extraordinarily slow reaction. What's wrong with this statement? Exactly, there can't be natural selection in the first place. So again, it's a curious blind spot from a leading enzyme chemist. Now, the last thing I just want to do is my field is chemistry. I know a bit about chemistry. So I want to just talk a little about this Miller-Urey experiment that you may have heard of. It's in all the textbooks, about how you have a discharge, you've got gases, a mixture of gases, and you have this discharge to provide energy to produce the building blocks of proteins. But in fact, it produces a mess, basically. It's a gunky tar. You need special equipment to separate the amino acids because it's jumbled up together. That is a real problem. And the other problem is, in the real world, you haven't got these traps here. You see, you've got traps here to protect the products from the destruction by the electrical discharge or ultraviolet light or whatever you want. You see, without the traps, the destructive processes would be about 100,000 times more than the constructive processes. So you have to have this, what is called illegitimate investigator interference. This could not be a valid simulation of life, of a primordial Earth, because it had too much of the intelligent investigator putting that trap there. So what can we say about this? Now, once we've got these building blocks, will they actually build anything? Well, not under the circumstances under which they're formed. You see, what we have here, to build up the big molecules, you need the little molecules joining together. And what we have here is called a peptide bond. These are the bonds between the building blocks of proteins. The amino acids join, and you see you've got something that will join. Both ends will join, and therefore you can get a protein chain. But it's not as simple as they make out. I'll show you what actually happens. Here's an animation we've got from Achilles' heels about how the amino acids are joining together. What happens though? They're spitting out a water molecule at every step. That's why it's called condensation. It's spitting out water molecules. The problem is these reactions are reversible. They can go the other way. As you see here, the water molecules can come back and break up the big thing into the little things. And therefore, any chemist who wants to make proteins has to get rid of the water. Water is the last place you'd want to do this sort of reaction. And yet they want to tell me that life began in a primordial soup in the ocean. Well, last time I checked, the ocean has water. So it really is the worst place you could expect to do origin of life experiments is water, because water would actually drive the reaction in reverse away from life. Now, another thing which is often overlooked is this problem of chain termination, because the good thing about the amino acid, they have something on both ends. You might say it's a bifunctional group molecule. It has one thing on this end, one thing on this end that can join to something, and so on. But in the Miller experiment, the most common product was formic acid, which is ant sting. Ant sting. Formica is Latin for ants, so formic acid is what you find in ant sting. So if you did an honest title for these papers, it should be a way of producing impure formic acid. That's what was produced. And the thing is, if formic acid is in that mixture, what happens is it reacts with the amino acid, but then it can't react any further. So the growing chain is therefore blocked at one end. And you also have chemicals that would block the other end. And there's actually a creationist that we interviewed in our magazine a while back, Dudley Irish, who said, I work in biotech producing a bifunctional monomer for the polymer industry. And I can testify to the fact that the purified material must be free of the monofunctional one. And in fact, how pure did it have to be? He said, the final product has to be greater than 99.5% pure. So 1 in 200 of the monofunctional thing would stop the whole thing from working. That's all it takes. In fact, he said greater than 99% pure. So it looks like 1,000 monofunctional things would ruin this whole thing. And yet, the Miller experiment produced about 5 times as much formic acid as glycine, the simplest amino acid. So 1 in 1,000 is enough to destroy, but he got a 5-to-1 ratio. So there's no way that you're going to get the proteins forming from the products of the Miller experiment. So what they have to do, they use the Miller experiment and say there's a trace of glycine and some amino acids. So let's go to MaxiPure Chemical Company and get some pure glycine and see what happens there. But they need, again, intelligent investigator interference to get the purified stuff instead of the dilute and grossly contaminated stuff that you get in the Miller experiments. So no chemist would do what these guys are proposing. So it's interesting that the primordial soup idea has been proposed dogmatically because they need it for evolution. And so Hubert Yockey, who's an information scientist, he said it was now the entire effort in the primeval soup paradigm is self-deception on the ideology of its champions. They believe this because they have to, because the only alternative is that we were created. And if we're created, the Creator owns us and has the right to make the rules for us. So what sort of things can we say about the resources here to sort of go into it? I mean, even my Genesis commentary talks a bit about this. You don't find many commentaries who talk about the origin of first life and information and design. Mine does. It's also the only commentary that talks about dinosaurs too, I think. Another one you might like is the video God the Master Designer. It has some of the animations that I presented here. And then you have the Evolution of Achilles' Heels. The book was written by 9 Ph.D. scientists in their own area saying what was wrong with evolution from their own specialist fields. And the video, which is really excellent for showing to Bible study groups or home groups or homeschool groups. It's got 15 Ph.D. scientists, and the video is in 9 separate sections about why evolution doesn't work. And some of the animations I showed you are in the Achilles heel DVD. So thank you for listening to me. So back to the boss here.
Excellent information. This is my favorite subject because this totally eliminates the possibility of evolution. Okay, it is 11:30. We're going to take a lunch break. We'll be back at 1 o'clock. So go fill your belly, stretch your legs, and we'll see you back shortly.